I want to spend a few moments talking about diffusion. This came in the end of chapter 6. It won't be on exam 1, but you're going to need to understand it before we move into unit 2. So let's take a look at diffusion, and before we specifically talk about what that is, we have to define something called a concentration gradient. A concentration gradient is when you have two sides of a membrane, and on one side you have a different number of particles than you do on the other side. We'll take a look at a graphic of this in a moment, but when a concentration gradient exists, particles will want to move because they don't want to be at two different concentrations. So they will move from high concentration to low concentration. And that is what diffusion is about, is that movement. It's a spontaneous event. Let's take a look at this. Here is a semi-permeable membrane, much like your plasma membrane is. And what you can see here in white, there's no movement allowed there. But in this little portion here, movement is allowed. You can also see that on one side of the membrane, you have these purple particles that are in high concentration over here. You have none of them on this side. And the same goes with the white balls, high concentration on this side and none here. So like we just said, because of that concentration gradient, because there's a different concentration or number of particles on this side than there are on this side, these particles are going to want to move from high to low. We look here, the purple ones will move from the left-hand side to the right, and then the white will move from the right-hand side to the left. That's what diffusion is. Eventually, you reach something called equilibrium, where the number of purple particles on this side is equal to the number of purple particles on this side, and the same thing for the white. That is called passive transport. It is diffusion, but it's passive because it's going to move without any energy source being put into the system. Now, there's a similar term called osmosis, and osmosis is basically diffusion of water. So let's revisit this here, semi-permeable membrane inside. You can see the green particles are in higher concentration on the right than they are on the left. But the other thing that I want you to consider is that if you have more particles, there's less room for water. If you have less particles, there's more room for water. So in this case, water is at a higher concentration, if you will, on this side than it is on this side. And water also wants to move from a higher concentration to a lower concentration. And that's what osmosis is. So you can see that in this case, the volume of water has gone down on the left and has gone up on the right-hand side. So the movement of water from a high concentration of water to a low concentration of water is called osmosis. If you put a cell, and this represents a cell here, the lipid bilayer, the phospholipid bilayer, if you put that cell into a solution that is called hypertonic, what that means is that the number of particles, any given particle, on the inside of the cell is lower than what's on the outside of the cell. So you can see lots and lots of the green balls in that hypertonic solution. What will happen is there will be a movement of water from the inside to the outside. Because remember, there's lots of water in here, high concentration, less water out here. And when that happens, the cell will begin to shrivel up and shrink because the water is no longer at a higher concentration inside. It's reached a balance. So in a hypertonic solution, a cell will shrink. In this case, they're calling it a vesicle, but your cells do the same thing. Now let's imagine that you have something called a hypotonic solution. Again, a solution is what is on the outside. And you can see here that we have less particles out here, more particles inside the cell. And what that means is there's more water here than there is here. So the water will want to move from a higher concentration to a lower concentration. And what happens is, you can see the water moving. What happens here is that the cell will explode, lyse, burst. Lyse is another word for that, L-Y-S-E. And if you put, let's say you put these cells into a solution of salt, where you had lots and lots of salt in the solution outside, your cell would shrink. And if you have it in water where there's not so many particles on the outside, the cell would explode. And if you imagine a red blood cell that's in this watery environment of your blood, you can imagine why sodium balance is so very important inside your blood. The last situation is what would be called an isotonic solution, and that is what we want to be represented because the number of particles on the inside matches the outside. Also, the amount of water on the inside matches the outside. And so the cell has what we call no net movement. There's a little bit of movement in, but the same amount comes back out. And so there's no change in your cell. Okay, again, here's our phospholipid bilayer. This shows you something that's also diffusion, but it's based on charge as well. So here you can see that we have chloride ions inside and outside. They're both at the same concentration outside the cell versus inside the cell. But what's different is the sodium ions. And you can see that this positive charge on the outside is outnumbering the positive on the inside by 6-1. And that sets up what we call an electrochemical gradient. So we have a high concentration of sodium on the outside, less sodium inside, and still the same amount of chloride. So you actually have negative on the inside. And here's that electrochemical gradient. And you will see the sodium begin to move from one side to the other because of that. Now, the last thing I want to talk about is when energy is required for these things to move. So we just talked about passive transport, passive movement down here, and that diffusion is passive. No energy needs to be put into it. It's based on a concentration gradient or an electrochemical gradient, and that means that these molecules want to move from a high to a low concentration, and they will do that without any help. Sometimes molecules do not want to move through this membrane, and they don't necessarily need energy to do that, but they do need help. They need a pore, and in this case you can see 3 different pores that are actually composed of proteins. And that protein will allow things like sugars to move into the cell. So this is glucose, which would actually sit down here, and then the pore would open and the glucose would move inside the cell. Water moves through something called aquaporins because water doesn't always like to move through that membrane. And then here's a potassium ion that needs help to get through. That's called facilitated diffusion. No energy, but needs help. And then the last one here is called active transport. And in active transport, I can move against the concentration gradient, but I need energy to do it. And ATP, we discussed, is the energy that we need to do that. Okay, so just as a recap, facilitated diffusion uses pores or channels. It's still passive. Substances still move with their concentration gradient. They're still moving from high to low, and it doesn't require energy. Active transport, remember, active moves substances against their gradient, which means from low to high, and it requires ATP.